Where do I do this thing?
Hofstadter Lecture. I apologize that there was some confusion about how to find a room this year. We had trouble getting the Hewlett building because of exams or some other reason. I can't remember exactly what that was, but most of you made it. I know a few had trouble finding the place. And Steve Chu, our distinguished faculty member, is going to do the introduction today of the Hofstadter Lecture, and Steve has promised that his introduction will be shorter than the lecture. So we'll try to get him hooked up and connected quickly. So we're very thankful to the Hofstadter family for providing support for this lecture series, and it allows us to bring some of the most distinguished scientists and speakers to campus, and it's a great opportunity for us again this year. So, you about ready? Yeah, I guess so. It wasn't so bad. Look at this. All right.
You've got the handful of dual screens. Instead of one nice big screen.
All right, so welcome to the Hofstadter Lecture. I'm going to do two things. Louder.
Once it's turned green.
Hello?
Once it's turned green, you can.
It's...
No, it needs to. All right, good.
Okay, thank you. See, everything has a delayed reaction. So, welcome to the Hofstadter Lecture. I want to do my job. I'm going to do two things just to remind people of what the Hofstadter Lecture is about. And it honors Bob Hofstadter. This is Bob, who was a very distinguished member of our faculty, a real pioneer in not only high energy physics, but in other things and applications of high energy physics techniques to biology, medicine. This is an early picture I was able to boot up of Bob on the left-hand side with Pete Panofsky and a young Doug Hofstadter in a experimental chamber that was being built that would eventually be able to look at electron scattering here at Stanford campus. And he, for those who know, he looked at electron scattering, and it was through this electron scattering that he was able to measure the scattering properties in light nuclei, and eventually the effective scattering radius of the neutron proton, and it was that work that he got his Nobel Prize. He was something, I also want to say that the Hofstadter family are here, and the Hofstadter family has been connected in very many ways. They also were great fans of Stanford, and for example, there's Bob, and you might think he's talking about physics, but really it's Stanford touchdown. Also, when I was there, I got to know not only Bob, but Nancy, who was also a dear part of the Stanford family. So anyway, so I think I'm not going to read all the things that's in your program. But so from that, I want to turn to introducing Xiaowei. And in introducing Xiaowei, here we go.
Oh, that can't be true.
She's very distinguished. came to the United States, became a graduate student at UC Berkeley with Ron Chen, and then after that, her advisor was raving about her greatly, and she, there's a thing called a Charter Fellowship, which is given to Stanford every couple of years from applied physics, and the idea is a very distinguished fellowship. You can get this, you can work with anybody you want, you can do anything you want, give you intellectual freedom, and so she got this charter postdoctoral fellowship at Stanford, and for better or worse, she decided to park in my group. Worse for her, better for me. And she stayed for 4 years. And she went on to go to Harvard after 4 years, and has won a tremendous number of awards. Most recently, the Breakthrough Prizes is an old thing. She just got, Sunday,
Yes.
a major prize from the National Academy of Sciences, and she said at this little thing afterwards, she says, I gotta go, because I gotta be at Stanford for a while. And so I'm very glad she was able to make it here and visit us as the Hofstadter Lecture. I should also say that in addition to these many, many prizes she won, all remarkably well-deserved, she has done many things in science. I'm not going to go through them, but one of the things which was spectacular was a continuation of what was happening in fluorescence microscopy, and that was a Nobel Prize shared by W. E. Murr, Stefan Hell, and Eric Betsy. And if there were 4, I think she would have been 4th. But there's only have 3, so Xiaowei, you're going to have to get your Nobel Prize for something else. That's okay. There's still time. And I also want to say, finally, that rather than list all the awards, I'm going to try to show you of some time when she was in my group. Now, Xiaowei's in the middle, and looked a little different. We all did. I was on the end. Bldg on the far right is now a professor at MIT. Taji Ka is now a professor at Johns Hopkins. There are many people in this, and also there was another then student in my group who used to make fun of me because I was so slow on the bicycle going up Omaha. And he was at least 5 or 6 minutes faster than me. And I was so proud, I could get up there from the bottom to the top in 25 minutes. And he was looking at me and saying, is that all you can do? And, well, anyway. So, Xiaowei and Hazen Bldg got married, have a child, and have been a team for a very long time together, professionally and also socially. And so those are other things that happen in research groups.
Hazen would like to speak.
Yes.
Yeah, Hazen's next to me. Doug Smith over there is Jing Chen, who's the newest chairman. Okay, so that was my group in those days. So I didn't take that much of your time, did I? All right.
You can take more.
No. But anyway, she's done spectacularly well, many things, and I don't really want to go into all of them, but I'm glad I'm stalling for time, because this place is probably hard to find. It's geometry, right? Okay, Xiaowei, thank you very much.
Yeah, it's entangled. All right. All right.
so you don't trip.
Okay. All right. So where is my this and the laser pointer? Is there a laser pointer? I guess no.
Can you?
Oh, I don't like, because I don't like to stay next to my computer. It's okay. We'll try to.
I have one.
Fellow alum from USDC.
Yes.
In Harvard.
And Harvard.
At Harvard, yes.
Stick together.
It tells you that people from Harvard have certain quality.
They're prepared.
Only some people from Harvard. The rest of us don't know how to operate a laser pointer. All right. Okay. Yeah, so, well, thank you, Steve, so much for the invitation and for the, introduction to make this all more of a relaxing thing. So then I can also just give a sort of a light mood after dinner talk without having to hurry up, right? Because also I know that if my lecture goes over time, nonetheless, it won't be as long as Steve's typical lectures. So he usually makes it twice longer than the allotted time. So I probably will be at most 10 minutes longer. So, right, the first, I really want to say that it's a great pleasure and honor to give this distinguished lecture in honor of Robert Hofstadter. And it's probably not atypical, because I actually, in preparing this, I told Steve that I didn't go to sleep until 3 a.m. last night because I was watching some of the past Hofstadter lectures and found that mine is very atypical because it's more biologically oriented, while the past Hofstadter lectures are all pretty physics oriented. So, I also would like to thank the Hofstadter family to be here. Thank you so much. And I remember in my old days, when I first started as a assistant professor the first few years, when I go around to give talks in the physics department, towards the end, there's often a question to say that, Xiaowei, what you talked about does sound interesting, but what's the new physics in it? So later on when I give lectures, I don't get that kind of questions anymore. I think that's because I more give lectures in the biology departments. So I really feel particularly honored to give this physics public lecture, and I would like to thank the committee members for selecting me. It shows what a open-minded committee this is, and they actually think of using the new physics tools to study biology as part of physics. So, to me, Stanford has always feel like whenever I come back to Stanford, it feels like homecoming, because I had 4 extremely good years here, and I had great mentors. I still have great mentors here, and many good friends. And so actually, one thing is, speaking of giving a more biologically oriented talks, I feel like Stanford is in part responsible. Where's my cursor?
No, it's on there.
Oh, I was trying to. Okay, all right. So I feel like Stanford is actually responsible for my switching to the dark side to study the messier biological problems. And Stanford, Steve was very generous. He mentioned that I got this Childrow Fellowship. He didn't mention how I got the Childrow Fellowship. So after I graduated as a physics PhD from Berkeley, I actually applied for faculty position here at Stanford, applied physics. And I probably got close, but in the end, I didn't make it. And the search committee chair, Professor Stephen Harris, called me and then he said, Xiaowei, we do like you here, but we discovered that you have never done a postdoc. So, and then he said, we would like you to do a postdoc. So hence, instead of a faculty position, we offer you a postdoctoral fellowship. Yeah, honestly, you know, I would say that at the time I got the phone call, it's, you know, one of the saddest things that I thought had happened to me. But now reflecting back, I feel like it's really, really, honestly speaking, one of the best things that has happened to me. So, you know, during those years in Steve's lab, I it opened my scope of, you know, research and knowledge, and then really after 4 years, I felt like I was much better prepared. I really knew what I wanted to do in my independent lab. So I say this because I see a lot of young, you know, students, whether it's undergraduate, graduate students, or postdocs. You know, doing a postdoc is a great thing, and being patient and accumulating your knowledge, and then get ready and open up your, you know, knowledge and research scope, and then by the time you're really ready to know what you do and start your independent career, that is important. Otherwise, if you get out there too soon under the pressure of you know, writing grants, publishing papers, you know, getting tenure and so on, it could actually narrow down the things that you want to do. And then also, you know, another reason why it's one of the best things that happened to me is I got to know Steve's lab, right? So, okay, that's a long-winded thing to say that, you know, why did I switch to biology? You know, I remember, you know, after it was the Charter Fellowship, like Steve said, I could choose any lab that I work with, and I talked around, and then I was really incredibly inspired by Steve, both his way of thinking and his research. So I decided to join his lab. But as a bold young postdoc, I said, Steve, you know, what you did in atomic physics and polymer physics is great, but I want to do something entirely new. And then I said, Steve, what could I do? And apparently I was clueless. And then he said, oh yeah, I got interested in biology, so how about biology? At the time, I really knew nothing about biology, so obviously it's new enough for me. So then I say, okay, why not biology? So that is actually how because you know, these days people ask me so much about how I transitioned to biology. You know, that's really the true honest answer. So not knowing what you get into is perhaps also a good thing, because I also don't know what the challenges lie ahead. But it's really, after I got into it, it's such a wonderland that I really never regretted of transitioning to study more biologically oriented stuff. So. With that, I would like to thank Steve for getting me interested in biology. But also at the end, if there is a question of saying, Xiaowei, what's new physics in your talk? Steve will answer for me. So as I mentioned, that when I joined Steve's lab, I really knew nothing about biology. I remember asking a fellow postdoc, hey, what is the difference between DNA and RNA? So he thought I was joking, but that was really, I truly didn't know. So then if there are graduate students or physics majors who are as ignorant about biology as I was, I decided to start with some simple facts. As we know that you know, living systems are made of cells, and cells are made of molecules. And in my lab, you know, these molecules, they really form intricate interaction networks, and then together give the cells behavior and function and life. In my lab, we're very interested in using direct visualization approach to understand these interactions. And that also had strong influence from Steve, because this is something that I remember when I was in his lab, he used to say it a lot. As Yogi Berra says, you can see a lot by just watching. So that has stuck with me for a long time. So we have been developing different kinds of imaging approaches to study biological systems and to directly visualize molecular interactions inside the cell. One of the challenges is the size of the molecules are small, because this is the microscopic size of us, and the cells are about 10 to 100 microns, and molecules are about 1 to 10 nanometers in size. And then if you wonder exactly how small that is, if you blow up a human being to the size of the Earth, then the cell would be roughly this size, Stanford campus. And then the molecule would be some object in this picture, and who care to guess?
Blade of grass.
Grass? Not that small. So, right, it's actually the head of a human being. So, so that actually, talking to other people, they might think it's really small. Talking to physicists, it's not that small, especially, at, Hofstadter Lecture, you look at the structure of protons and neutrons, you say, well, this is a huge. But imaging biological system has its own unique challenges. And that's because inside the cell, we're not talking about just one kind of molecule. There are actually thousands of different kinds of molecular species that are collectively interacting and functioning to give the cell its life and behavior and function. So in addition to be able to see these tiny molecules and have molecular scale resolution, we would also like to have what we call molecular specificity, means to know the identity of the molecules. And then also because, cell or living systems, they're live objects, so they're constantly undergoing changing and motion and dynamics and so on. So we would also like to have dynamic imaging capability. So if you look at these three requirements and then think about what kind of microscope does pretty well, and then you quickly think about light microscopy, and in particular fluorescence microscopy. And that's because, of course, being light microscopy, we know it's compatible with living systems. After all, we go out to the sunshine and get sunbathed and without getting hurt too much. And the molecular specificity often comes from labeling, because first, light has so many different colors, and second, there are so many different colored fluorescent probes, and these could be natural biological objects such as fluorescent proteins that you can link to the biological molecules of interest with specificity by genetic approaches, or it could be things like antibody that not only are responsible for our immune response, but can be used to recognize specific protein molecules. And then there, you could also put fluorescent dyes. So with these labeling approaches, you can light up specific biomolecule of interest and study that. And what light microscopy used to be not so good at is actually on the size front, meaning the resolution front. And it doesn't have the molecular scale resolution. And that to physicists is a well-known problem, because the light is a wave, and as all waves, they diffract. And then in the microscopy world, the diffraction manifests itself in a way that no matter how good an objective you find, and then you try to focus your light spot, the light spot's focal spot size nonetheless, the focal spot nonetheless has a finite size. Even with the best objectives, that is about 200 nanometer in this transverse direction and about a micron in the longitudinal direction. That means any tiny object, even a single molecule, when you image it, its image will spread out into a finite size. And if you have two molecules that are well separated, then of course their images are separated and hence resolvable. But as the molecules get closer and closer together, eventually you would have them get into a distance that's comparable to the size of their image, and their images will be tremendous or substantially overlapping, and you can no longer resolve them. That was actually recognized by Ernst Abbe in the 1870s, hence this diffraction limit is also called the Abbe resolution limit. And of course now it's well known that this resolution limit is no longer a limit, because it has been overcome, and it actually has been overcome optically by multiple different approaches, and there are actually two major categories of approaches, and one of them is using pattern illumination to effectively shrink the point spread function. Point spread function means if you have a single point, you image it, it'll spread out into a size. You effectively shrink that point spread function into a much smaller size, and a very famous pioneering example of that is the stimulated emission depletion microscopy STED, and then also later on it's more generalized form resolved. Both are pioneered by Stefan Hell at Max Planck Institute. And what I will talk about is the second category is single molecule imaging based, and that's what I will primarily focus on today. And so let's get to single molecule approach. So as you at Stanford should all know, that W. Moner sitting here actually demonstrated the first detection of single dye molecules in 1989. And now once you can image a single molecule, even though the image of a single molecule is diffraction limited, means its width is about 200 nanometers, nonetheless you can pinpoint the center position of this image by a much higher precision. And that precision is roughly the image width divided by the number of the photons that you detect. And that is very easy to understand because you can just consider every photon you detect a single measurement, and that measurement has an uncertainty which is defined by the width of the point spread function. If you detect 10,000 photons, that's as if you have done the experiment 10,000 times. And in freshman physics, we know that how to reduce the error by doing many, many experiments that reduce with a square root relationship. So you take 10,000 square root, that gets to 100, and then you take 200 nanometer divided by 100, that gets you 2 nanometer. So that itself is actually a very powerful approach already. For example, Paul Salvin in this paper actually demonstrated that you could see this hand over hand. He did actually localization with 1 nanometer precision, and then you can see this hand over hand movement of an amazing motor protein called myosin. And then I was talking to Steve today, and then it's really amazing that Steve's lab has now developed these never die, I don't know whether they really never die, but very, very long-lived rare earth nanoparticles that could give light but live really, really long. And with that, you can track biological molecules, and then Steve has used it to learn not only amazing biology, but also amazing physics. So, but as I said, the resolution problem comes from when molecules or objects are too close. When they're too close, their images overlap. And when they overlap, you can no longer resolve them. So this overlapping problem is actually the fundamental cause of the diffraction limit. So how do you solve that problem? And that can be very severe. If you look at a biological specimen, we're not talking about just 2 molecules. We could have thousands, if not millions of molecules. Their images overlap. And then there we lose the ability to pinpoint their center position. So how do you solve that problem? That's like really now more than a decade ago, we and others had come up with the idea of using what we call the 4th dimension or the time dimension to solve the problem. The idea is we do not turn on all the molecules simultaneously. We turn on a subset of the molecules simultaneously, such a small subset so their images no longer overlap. And that allows you to pinpoint their center positions. And you turn these off, you turn on a different subset, you pinpoint their center position, and then you iterate this thousands, if not tens of thousands of times until you determined all the molecular positions in your field of view. And then when you put all the positions together, then you can reconstruct what we call a super resolution image. And that image resolution is no longer diffraction limited, means not limited by the width of these single molecule images, but by how precise you can localize them. And prior to we developed this approach, what led to us to develop this approach is actually we'd identify molecules that would allow us to do that. And these are the photo-switchable dyes, and they're actually separated by free energy barrier. So if you use a weak enough light to activate these molecules, only a stochastic subset of them will be able to go across the barrier into the fluorescent state, and that allows us to turn on only a subset. So that's why we call this approach stochastic optical reconstruction microscopy, hence the name STORM. And as many of you know, that they were independent parallel work. Eric Bldg, Harold Hess, and Jennifer Limpenkow-Schwartz, they developed a similar approach called PALM and published simultaneously to STORM. And Samuel Hess independently developed a similar approach called FPALM and published just slightly later. And then here is sort of how we do this kind of experiment. You activate a subset, you pinpoint their center position. After enough time accumulation, this is the kind of image that you can get. This is a structure called microtubules inside the cell. And this is, in comparison, the conventional diffraction limited image. So even without quantification, you can directly visualize, see how much resolution gain we have. I'll give you numbers just slightly later. But I want to jump into the next thing because soon we realized that it's not sufficient to overcome the diffraction limit only in 2 dimensions because the biological structures are mostly 3-dimensional. So we need to overcome the diffraction limit in all 3D. Now in the STORM context, conceptually it's really simple because we did the 2D super-resolution imaging by determining the XY position of the molecules. We just need to add the Z coordinate. Now it's slightly easier said than done because you take cameras to take your images. These are just flat images. How do you get the Z position information? But if you play it with microscopes, you probably all realize that there's this focus knob. And then this focus knob allow you to put things in focus or out of focus. And then if you have a molecule in focus, it gives you a sharp image. And then when it's out of focus, it's blurry. What that means is actually the shape of the image encode the Z information of the molecule. But that itself is not really sensitive, especially for physicists. We know that we're sitting at a local minimum, and below and above focus, that it has even degeneracy. So to solve this problem, we insert a cylindrical lens into the detection path, and cylindrical lens to look like that. And this is something that some of you probably are familiar with, because if you have astigmatism, then your correction eyeglasses actually is the cylindrical lens, right? So that only focus light in one direction, but not the other. So as a result, the single molecule images turn elliptical, and the ellipticity depends very sensitively on the Z position of the molecules. And below and above focus, they are elongated in perpendicular directions and break the degeneracy. So with that, we got this kind of a 3D super resolution image, again, STORM image, again, about microtubules, and now you can take any area and then you can rotate them around and then you can get the 3D view. And then here's another such example of a different biological structure. And I typically do not show this image, but here I show it because, when news reporters ask for me to give them STORM images, they almost always select that one because they say it looked like a galaxy. So then I thought, okay, that may be the closest I get to astronomy. So with that, you can see that this is the conventional image. This is the 3D STORM image with the color coding Z, and this is a cross section. You can see this tubular structure that you cannot resolve with conventional image. And then in this original 3D super resolution imaging work, we got about 20 nanometer in XY and 50 nanometer in Z. And then with that, it's about 10 times better than the diffraction limit in all 3 dimensions. And then I should say that there are multiple different ways of determining the 3D. Astigmatism is a simple way, but for example, W. Moner has developed this PSF engineering approach to turn this wavefront into this really cool double helical structure. And then from the double helical structure, you can actually, and later on single helical structure. And corkscrews, he likes to play with wavefront engineering. So, and then there are also interference approach to get you even higher Z resolution than these, shape of the wavefronts and shape of the point spread function and so on. So was this 10 times better resolution, that was at a junction that we felt like, there are really two kinds of things you could do. It depends on who you are. And if you are a biologist, you look at it and say, wow, I have 10 times sharper vision looking into the cell. I could use that to make a lot of wonderful biological discoveries. And for a physicist, you look at that, you say, well, you just told me that the molecular scale resolution is just a few nanometers, and this is a few tenths of nanometers, clearly not really at the molecular scale. So then you would want to further improve the resolution. So I will not focus on the improvement of resolution, but on the biological discovery. But I want to say that indeed, over the years, our lab, other labs, many labs have been improving this. I just list a subset of our work that you can basically improve the resolution by either improving or advancing or developing new optical schemes or advancing the molecular probes to make them emit more photons. And with that, indeed, we can now have a few nanometer resolution, which is truly at the molecular scale. And also STORM and other super resolution light microscopy approaches, they do inherit the light microscopy merit, and that is they are live cell compatible. They can be used to image living systems to get dynamics with both high spatial and temporal resolution. And of course, we know that spatial and temporal resolution is always a trade-off. So getting higher resolution, we do get slower speed. But nonetheless, even in this early work, we get pretty fast temporal resolution with spatial resolution 10 times better than diffraction limit. And then it has been improved since. So with that, and let me turn into the biological discoveries. Another thing that super resolution light microscopy like STORM that inherit the merit that inherits from light microscopy or fluorescence microscopy is it can be very widely applicable to many different biological systems. And here I list only a subset of our biological studies that give you some idea of the wide range, because it really ranges from really simple systems such as bacteria, where we could see in this work every individual ribosome molecules in an E. coli cell, to this another relatively simple system, which is a sperm tail, where this is actually a collaboration with David Clapham's lab, where we looked at an ion channel called Casper, and then found that this ion channel formed this really amazing quadrilinear kind of a domain structure, which David's lab later on shown to be important for the hyperactivity of the sperm, which at least will make half of the audience happy. Maybe not even half. I mean, not limited. So sorry, let me not go there. Right. So that's why I was hesitating when they were asking me, can we record your lectures? So, all right. And then ranging it all the way to complex biological tissues, such as brain tissues. And this is a nice collaboration that we've done with my colleague Catherine Bldg's lab. We look at the neuronal synapses and see the subsynaptic molecular structure. And since then, we actually extended to other synaptic proteins. This is a very nice collaboration with Tom Sudol's lab, where we look at a synaptic adhesion molecule, neurexin, and then see it form this really intriguing tight clusters in the synapses. And all the way from such small structures to large scale, we can actually use it to look at neural circuits, to see how neurons are connected, and to look at each and individual connections, which are these synapses, and to see what kind of connections they are. And then also even look at things in the pathological tissue. And this is a more recent study where we looked at a extracellular matrix called perineuronal net in collaboration with the Takao Hansch lab at Harvard MCB. And then we actually saw interesting difference in structure, both in terms of this perineuronal net and how it interacted with synapses and so on, in an autism model, actually in the Rett syndrome model where the MECP2 molecule is depleted. And we can not only look at structures in the cell has the cytoplasm and then also has the nucleus where it actually compartmentalizes our biological, our genetic material. We can not only look at structures in the cytoplasm, but also look at how DNA is folded in the nucleus and so on. But in terms of, actually here, maybe I'll expand that a little bit to say that, it's interesting because you can actually think about the DNA as a physical system because it's a polymer that's folded up into three-dimensional structure, and this three-dimensional structure is very important actually for its function. So there was an earlier work, we actually looked into how their folding changes depending on whether they're transcriptionally active, inactive, or specifically repressed with a mechanism. This polycomb repression is repressing those genes that should be turned on during the development, but should be turned off. Turned on during the development, but rigorously turned off afterwards. So you really don't want them to be expressed. And then we look at how they are folded by looking at the three-dimensional size versus the linear length. And then you can see that for the active DNA, it follows a parallel scaling, which is actually quite similar with this scaling exponent to a random polymer. And then if you look at inactive chromatin that are not actively transcribed, the scaling exponent actually reduces. And if you look at this compressed one, it further reduces. And then also the compressed one has a smaller size. And one of the reasons that I specify this work is because the first author who did this was a postdoc in my lab and now a faculty member actually at Stanford. So he really likes this be smaller than one scaling, because it's really intriguing. It means the bigger the domain gets, the more compact and tightly packed it is. So he used to refer to this as a magic suitcase that you can actually pack it well. But I think the reason that he likes it so much because it also has a strong biological implication, because this compaction is actually a good mechanism for preventing the transcription factors and polymerases from getting there to induce transcription. The better it compacts, the stronger that is. And that could actually explain that these polycomb repressed genes tend to concatenate together because it gets them longer to compact actually more. So these are just some anecdotal or quick snippets, but I want to actually focus on one discovery that we've made by super resolution imaging, because with this, it actually demonstrates that with super resolution imaging, we can not only get sharper and prettier images, we can even discover structures that we didn't know existed in cell before. And that has to do with a skeletal structure that we discovered in neurons, and that started with our studying of actin. So let me give you a little bit background. In cells, there are these what we call cytoskeletal filaments. Actin is one of them, microtubules, neurofilaments, intermediate filaments, and so on. So in a typical cell, this is what you see the actin filament look like. They form these long filaments and then crisscross into network type of structure. And this is actually a super resolution storm imaging image was 10 nanometer resolution. At that time we got that, we were very happy because it's the first time that actin filaments inside the cell are optically resolved. But I also have to be honest that there's nothing new about biology in this image. But what surprised us and everybody else is when we applied the same storm microscope to look at actin in neurons, and there we discovered an entirely new kind of a structure. In addition to these kinds of long-acting filaments, the most prominent feature that we see, I should say that for those of you who do not study neurobiology, neurons have two kinds of processes, dendrites and axons, and the signal propagates from axons to the target, the dendrites of the target neurons. So in axons, we see these kinds of a structure, the periodic band-like structure. And it's actually, this is a 3D storm image, so this color actually doesn't indicate different real color. It's actually false color for Z, which means we can turn these stripes around and then see this ring-like structure. So these are these little rings that are wrap around the circumference of the axons right underneath the axonal membrane. And then why are they missed? Well, this is why, because if you turn on just the diffraction limited optics, the periodicity of the structure is really just below the diffraction limit, so that it completely blurred out and then the structure is missed. And the periodicity is really, the regularity is really amazing. It's one of the most regular biological structures that I've seen, because if we could actually project these things onto one dimension and do a Fourier transform, you can not only see the sharp peak, you can see the multiple overtones and so on. Or if you take autocorrelation analysis, you can see this really periodic structure. And this periodicity is about 180 nanometers, as I said, just right below the diffraction limit. But actually the number, this 180 nanometers, actually give us some clue of what else might also be in the structure. Because if you look at this periodic band, you couldn't help thinking that maybe there's a molecular spacer. And this molecular spacer, if it exists, needs to satisfy two requirements. One is it needs to be about 180, 190 nanometer long. The second is it needs to interact with actin. Now, with that in mind, that got us to this biological molecule called spectrin. And the best that we know about spectrin is in an entirely different kind of a cell called red blood cell. Underneath the red blood cell membrane is this kind of a triagonal or hexagonal kind of a network structure. And the spectrin tetramer is this red thing, is forming the arms or the edges of this network. And that's about 190 nanometer long. And also you can see here, it directly or through another molecule interacted with this little green filament, which is actin. Hence, it actually satisfies both of our requirements. Now, with that in mind, then we hypothesized that the structure that we saw are actin rings formed by these short actin filaments, and then they're periodically spaced along the axons by the spectrin tetramer. So with that in mind, of course, we should test this hypothesis, and then a direct way to do that is by doing multicolor storm imaging. Now, if we do multicolor storm imaging, imagine that we can not only image actin specifically, but also image spectrin. Not only spectrin, but also the middle portion of spectrin. And then we should expect to see alternating green magenta rings. That's exactly what we saw. And then, maybe I skipped this part. We also found these little actin filaments to be capped with capping protein adenosine. And then we found later on many proteins to be on the structure. I only have time to mention a couple. That, for example, it has adapter molecule that actually link interesting membrane proteins such as ion channel into the periodic structure. So we actually give this structure a name. The name is not something that I'm so proud of because it's very boring. It's called membrane-associated periodic skeleton, MPS. So the name actually has a story. So we didn't intend to, in our first paper, we didn't actually name this structure. So then one day I was having I was actually at this, W and I, we were both on this Welch SAB meeting, so it yearly gives this great Welch Chemistry Prize. So I was attending the banquet of this Welch Prize, and then sitting next to me is another SAB member, Joe Golston, which is an extremely distinguished biologist. So he asked me, he said, Xiaowei, what's your new discoveries these days? So I described the structure. He said, oh, that sounds very exciting, but you got to give it a name. If you don't give it a name, how could people remember that? I said, good, let's think about name. In the lab, we call it periodic membrane skeleton. PMS. So that didn't sound great, so then I turned it around and then called MPS. So from now on, I'll refer it to MPS. So later on, we actually had a much complete picture of what the MPS structure looked like in neurons. In axons, it formed this highly periodic grain-like structure. And then we found that also in dendrites, it can form these periodic structure, but in a fraction of dendrites. And that was in this later on paper. And also Stefan Hell's lab also saw this periodic structure in dendrites. And then we also noticed that, in the portion where the dendrite do not form the periodic structure, it actually looks like pretty regular too. And then this looks like a hexagonal lattice structure, and which is even more pronounced in the cell body. And here I show you. So now we have a pretty complete, and then in the cell body, this lattice structure actually now resemble the red blood cell structure. And then this periodic ring structure forms much more slowly with less propensity than that in axons and so on. So it's a very prevalent structure, and it can be manifested in a variety of ways. For example, this is a whole field storm image. Every single axon, this is a dendrite in the middle, every single axon shows this periodic structure. And then later on, we actually looked at that into a variety of different kinds of neurons, and that include excitatory neurons, inhibitory neurons, neurons in central nervous system, peripheral nervous system, neurons that myelinated, unmyelinated, they all have this structure. And then independently in the same year, Stefan Hell published a paper which studied a overlapping but different subset of neurons and then saw the same thing. Basically, as far as we could tell, all the neuronal types that have been investigated have this structure. It's also a highly conserved structure because we originally discovered this structure in mouse and rats, and then later on we saw that it really, in a variety of animal species, ranging from C. elegans all the way to human. So being so prevalent, it suggests that this is a functionally important structure. So what kind of a function you could have for this structure? And I should say that there's a variety of functions that begin to emerge, but I suspect that that's still just a small subset. And one of them has to do with the property of a spectrum being very flexible and the acting ring being rigid. And it reminds us of this thing, which is a vacuum hose, but I often demonstrate this. If you take a rigid ring connected by a flexible thing, then what can happen is you can remain the integrity of this tube while exerting mechanical stress without breaking it, okay? And that is actually not just imagination, because experimentally it has been shown by the Jurgensen and Michael Bastiani lab that if you actually knock out beta spectrum, which is this purple thing, from C. elegans, what you see is the axons tend to spontaneously break. And interestingly, if you grow the animal, paralyze them so that they can grow but not move, the axon actually grow into fine shape, which tells you that this MPS structure is there to protect the axon under stress and then to be important for its survival. And then we also, as I said, we identified a variety of different membrane proteins on this structure, and then that indicate or suggest that it is important in axon signaling, and I'll show you a little bit of that in one story. And it's also been found to be critical for the formation of two important structures on axon, which is called the axon initial segment and nodes of Ranvier. Both of these structures are important for the generation and propagation of action potentials along axons. And in a more recent study, when the structure is disrupted in animals by Matthew Raspin lab, they actually see the mice exhibiting a variety of neuronal disorders, such as ataxia. So as I said, there are also implications in both healthy neuron development and in diseases. This is actually a recent study that we have in collaboration with Mark Tessier-Levine's lab, and that the work is coming out in eLife very soon. And it's really a long story that I won't have time to go through all the details, but just to give you the final conclusion that started from our observation actually first back off a little bit on axon degeneration. Axon degeneration is a phenomena that is both for healthy neuronal development and implicated in diseases. For example, in healthy neuronal development, you first grow out many more axons than finally you would need. And then depends on where the neurotrophin is. If you have the neurotrophin, the axons there actually can survive. If you are deprived of these neurotrophins, the axon actually degenerate. And then, as I said, there are pathological conditions that cause axon degeneration too. So here is a neurotrophin deprivation to show you that once you actually remove neurotrophin over time, you can see the axon degeneration. And Mark's lab actually has figured out that this actually requires both actually signaling, the retrosignaling from the axon to the cell body, transcriptional response, and then anterograde signaling from the cell body to the axon, and so on. So to make a long story short, I would just mention two things. That we found that together with Mark's lab that this periodic MPS structure quickly disassembles once you deprive the axon of the neurotrophin. And originally we thought, oh, that's just some expected result, because once the axon degenerates, the structure, of course, will disappear. And then especially because once axon degeneration happens, you have these caspases that are cleave proteins that could actually cleave these structures. But what's amazing is we found that this structure actually disassembles before the caspase activity gets turned on, actually in a manner which is entirely independent of the apoptotic pathway activation. And we can even genetically block the apoptosis, and yet the structure still disassembles. And then we found that if we destabilizing actin, which cause MPS to disassemble, sorry, this is not disassembly, we could actually induce this axon degeneration or retrograde signaling pathway. If we stabilize actin, which prevents MPS from disassemble, we can also prevent retrograde signaling and axon degeneration, which shows you that the actual disassembling process of the structure is important for axon degeneration. And if that's the case, if you completely remove the structure, you should also not have axon degeneration. And indeed, if we actually genetically deplete beta-2 spectrin, you can see that normal axon would degenerate, and then in this beta spectrin depleted neurons, degeneration no longer happen, and retrograde signaling no longer happen. So what we think is this MPS structure actually anchors signaling molecules that are important for retrograde signaling, and once the neurotrophin gets deprived, it releases these signaling molecules, and perhaps that way start the axon degeneration. As I said, I have to make this story really short, but it has a lot of more details in it. But I want to end this part by saying that, of course, we're not the only lab who does super-resolution imaging. Super-resolution imaging, both in terms of technology development and biological applications, has been done by by many labs. And here is a recent review article that we actually wrote to cover some of the technological advances, but more focused on the wide range of biological applications. W here, I have your beautiful parade structure in this picture. So with that, it's really beginning to give us a much better, clearer, and more detailed picture of what's inside the cell, the molecules and their interaction networks. And the hope is with the development of imaging methods with a higher resolution, but not only higher spatial resolution, but also higher temporal resolution. And then eventually we would be able to see the molecular events unraveling right in front of eyes, just like this kind of a movie. And to illustrate this point that Steve mentions again and again during my postdoc period, that one can see a lot by just observing. So one thing that I should pause here to say that, here I want to emphasize one other aspect to let to, the last several minutes of my talk, which is primarily sort of a prelude to my talk tomorrow, to say that, which is the thing that I've already said, inside the cell, we're not talking about just one kind of molecule, but many, many, many different kinds. We're talking about thousands, tens of thousands of different kinds of molecules. They're interacting together, collectively give the cell its behavior and life. So in addition to these wonderful molecular scale property, molecular specificity, and dynamic imaging capability, it would be really wonderful to also have another property of imaging, and that is the whole genomic scale imaging. And that would give us sort of a systems view or genomic view of how cell functions and so on. And there I'm going to just very briefly cover a few things, but primarily going into more details tomorrow. And with that, along the central dogma of biology, DNA gets transcribed into RNA and then gets translated into a protein, we started this genomic scale imaging effort by starting with RNA. Okay. And that choice is actually quite obvious for both technological and biological reasons. Technologically, you'll see right away because the labeling is relatively easy. Biologically, the way to think about it is, inside our body, we have many, many cells. These cells, there are many, many different kinds. And these different kinds of cells are not because they have different genome. They by and large share similar and often identical genome. And the reason they're different is because their genes are expressed at different levels in different kinds of cells. And RNA, of course, is the first step of gene expression. So if you could profile RNA, get the gene expression profiles, and then you stand the chance of, learning a lot of hypotheses about the, molecular basis of different cell behavior and function. So how to do that? And we know that imaging is not known to give this genomic scale. Actually, if you think about the kind of imaging experiments we typically do, we do one color imaging for one molecular species, 2 or 3 color imaging to study the interaction between 2 or 3 different color species. And the genomic scale means thousands, if not more, different genes. And the problem is we do not have thousands or more distinguishable colors. And that is a very well-known physical property, and that is because these fluorescent molecules that we study, they have finite spectral width. And then these finite spectral widths make it only possible to distinguish a handful of different kinds of different colors in the visible wavelength range. So how do we solve this problem? And we come up with this idea of error bus barcoding, combinatorial labeling, and sequential imaging. That might sound complicated, but actually it is not. Let me just briefly explain. So what we do is for each individual gene or RNA species, we assign it a binary barcode, a distinct one for distinct species. And then we physically imprint the barcode onto the RNAs. And then we read out this barcode 1 bit at a time. And the first round of imaging, we read out those barcodes. Their first bit reads 1 but not 0. And the second round, we read out those barcodes. Their second bit reads 1 but not 0. Now, how do we do that? Labeling and reading. We use an approach called single molecule fluorescence in situ hybridization, which is pioneered by Rob Singer and further developed by Singer and Arjun Raj and others into a highly accurate approach. What it does is it uses complementary oligos to identify RNA molecules, and these complementary molecules are labeled with fluorescent dye. But for our imprinting, we actually come in with this complementary oligos that has a special property, and that is they have these readout sequences. And these readout sequences are exactly designed based on their barcode. If it's a 16-bit barcode, we only have 16 readout sequences. If it's 1010 and so on, they only have readout sequence 1, but not 2, 3, but not 4, and so on. And then we come in with the readout probe that's complementary to the readout sequences to read out which readout sequences are there. And then in the first round with the first readout probe, we now address only those genes that their first bit reads 1 but not 0. The second round, we do the genes that their second bit reads 1 but not 0, and so on and so forth. So at the end of imaging, what you get is for each individual cell, you have a bunch of dots in it, and then each dot has a binary barcode associated with that. And a very amazing property of this kind of imaging is how it scales. Because, if I were to say that I don't have 10,000 colors for you to do 10,000 genes. And you can say, well, if I'm sort of patient enough, I do one gene at a time. I label the first gene, I remove the probe. I do the second gene, I remove the probe. And eventually, after 10,000 times, I also imaged all of them. And of course, I couldn't convince any of my students to do that kind of a patient experiment. And I don't think any cells can survive that way, too. But the good thing about this is after n-bit imaging, how many different molecular species you can distinguish that grows exponentially with the bit. So just a 16-bit gives you 60,000 RNAs. Okay, so that's actually basically the whole human transcriptome size. I will get into more details tomorrow, but the one important thing about that is there's one caveat. This caveat, which essentially says what I just said here is a bit too good to be true, because there's error in every kind of measurement. Okay, I said the single molecule fish can be extremely accurate. Let's say if it still has 5% error left. For imaging one kind of RNA molecules, that's of no consequence. But that means every bit has 5% error. Now 2 bits has 10% error, 3 bits has 15% error. They add up. So you can quickly misidentify molecules. So how do you solve that property? And here in Stanford, sitting next to Silicon Valley, where people talk about ITs, information technology, we know that the information these days, whether it's text message and so on, they're all transmitted with binary barcodes. And we rarely get any garbled messages. That's because only error-robust barcoding are used. And in a nutshell, that means we don't use all possible binary barcodes, but a subset of them that distinguishing valid pairs of barcodes are multiple errors. And this way, you dramatically reduce the possibility of error. As I said, that's primarily the topic of tomorrow, so I'm just going to say that with this, we call this approach multiplexed error-robust fish, and indeed use that we can image thousands of different kinds of RNA species in individual cells. Now with that, I want to just mention there are a variety of applications of that, and one application that I will focus on tomorrow is actually going back out of this level, but to this level. Think about the kinds of cells that are in our body and how many different cells we have in our body is a pretty amazing number. 30 to 40 trillion cells in our body. And how many different kinds of cells are in our body, that we still don't know. Okay, so that is really an ongoing major biological question. But think about why, because if you think about previously, how do we identify cell types and so on? It's like, measure the shape, measure the function, measure the property. But where do the properties come from? different gene expression profile. So if we could profile individual cells, their gene expression, then we can have a more quantitative and systematic way of identifying cell types. And moreover, of course, there's sequencing-based approach, but if we can do it with an imaging-based approach, which I just showed you that our imaging capability allows us to do that, what can you do is in our body, these cell types, they're certainly not randomly distributed. Just imagine that if you have 1,000 different types of cells in your body forming amazing different properties and functions, and yet they're homogeneously randomly distributed. And Steve would definitely not look as intelligent as he is with that kind of a random distribution. So the spatial organization is really important for tissue function. And if we could use the imaging approach to identify the cell types, then we directly get the spatial organization and function. And indeed, we did that with a brain region where we see beautiful different types of cells and their spatial organization in the brain and their function. So if you're interested, I will share with you tomorrow. And then just to show you how dramatically this kind of a spatial organization can change, 0.5 millimeter more anterior is a totally different kinds of spatial organization. And with that, you could imagine that with this approach, we can now begin to think about building the cell atlas of human and other living systems to find out how many different types of cells are there in our body, how are these cells organized, what are their functions, how do they communicate with each other. And of course, we can not only apply this approach to healthy tissue, but also pathological tissue such as tumors to look at microenvironment of tumors, immune cell tumor interactions, and so on. Again, what I tend to tell people is it's just like building a Google Atlas of our body, but not only knowing what types of things there, cells there are, where they are, but also what they do. So with that, I see that I used up an entire hour. I was having 2 slides about the genome organization but it's again an introduction of tomorrow's topic. So I could also stop here, or I could just use 2 slides to introduce. So basically the genome inside our cell is actually folded into a really intricate 3-dimensional structure. And the 3-dimensional structure is more and more recognized to be very important for regulating a variety of genome interaction functions such as gene expression, DNA replication, and so on. So the multiplexed FISH approach that I just mentioned, you can imagine that if I do not just apply to RNA, instead of using multiplexed FISH to identify different RNA species and localize where they are, I used it to identify different genomic loci to localize where they are. We can then trace out a precise 3-dimensional structure of the genome, which is also something that I plan to talk about tomorrow. So with that, you can imagine that we can triangulate to actually look at the whole chromosome organization, to look at the whole genome expression profile, to understand how the 3D structure, the 3-dimensional structure of our DNA polymer in our cell or regulate gene expression, and how gene expression affect this 3D structure, and how the gene expression influence cell function, and so on. So with that, let me actually end the talk by thanking the people who has done the work. So it's I've covered a lot, so it actually would take too much time, I think, to thank all my former and current lab members who are even just involved in the work that I covered. These are the bolded ones. They would also take too much time. So I apologize to my group members, but in terms of, in the interest of saving your time, so I won't go through each individual one of them. But I want to say that it is really I always say that working with these super talented and dedicated young scientists is the most rewarding experience that I've ever had. So it is really their work and dedication that led to all this. And I've thanked my collaborators along the way, and then I think it's just in the end when I talked about the cell atlas work of a brain region, I didn't mention the collaborator. That was my colleague and friend Catherine Bldg, but I will certainly highlight her contribution tomorrow. And other collaborators, I think, of the work that I mentioned today, I did actually emphasize those during my talk, and the work is supported by Howard Hughes and Howard Hughes Medical Institute and NIH, and I thank you very much for your attention.
It's late. Would you be willing to take a few questions?
Sure.
Okay. And so open up the floor.
You mentioned trying to understand how cells communicate with each other. But though I would imagine would happen fairly rapidly, so how is it that you would be able to image, say, chemicals going from one cell to another with the techniques that you have?
Right, so I did mention in the cell atlas part, of course, there's not a single imaging method, or even just imaging methods alone wouldn't be able to answer the question. You would need even also other approaches. So what I meant by communication is, for example, you would have in this brain work, we often see hormone signaling, and between neighboring cells, you would actually see hormones released and hormone receptors in neighboring cells and so on. So that gives you implication of signaling modes and networks and so on. And similarly, if you look at in cancer microenvironment to look at how the immune cells interact with healthy cells and tumor cells and so on. But you're absolutely right that just a fixed picture like this is not going to give you all the answer to how cell-cell communicate, but it can generate hypotheses that we can further test.
I'm going to ask a question. I learned or heard that when we're very young, we're hugely connected brains, and the first order of business is to get some order to cut open.
The pruning, yes.
Which it sounds like that was part of these connections.
Yes.
That you're doing with our president. And so you're getting the molecular signals that are doing that, but how do you decide, in the order of pruning? Because you're wildly connected. If you have too many connections, you can't make heads or tails. So what's the hypothesis for, in these young babies, how do they decide what to prune? Or is it just, experience? Or whatever that means.
Yeah, I wish Mark is here, right? So, of course, he's so busy. So the presence of neurotrophic factors is part of the answer of, if you have the neurotrophic factors there, then those axons won't be pruned, right? And then if you,
it's...
Right, so, but that's only part of the story. And there's indeed also the learning process. Also not only axons have pruning, and dendrites has pruning too. So you're right in a sense that there are connections that could be originally built and then eventually get pruned, and then processes and connections that get strengthened. That also depends on learning and an experience. I think I'm actually a little bit speaking out of my depth, so this is not an area that I know very well. Yes?
So I want to ask my favorite question again about the MPS.
Yes.
These periodic patterns. Now you found periodic patterns in ion channels as well. And I want to know if there's been evidence found to show that this is essential for the action potential propagation, which is a wave of depolarization between potassium and sodium all along the axon. Isn't this just the right scale to make that work very well? Because you can't depolarize the entire membrane. You have to only depolarize it locally.
So the answer to that is, this is a question that we've been interested in for a long time, but to answer it biologically takes quite some preparation. So we're gearing up the preparations to do that. And then along the way, we've discovered the functions of this structure in other axon functions. And the preparation that what I'm referring to is you want to perturb the structure rather specifically. and see how action potential gets perturbed, okay? So if we actually perturb some of the perturbation that we have, such as disrupting the actin, remove the entire structure, or knock out the spectrum, remove the entire structure, it not only gets, it does have influence on action potential pattern, okay? So, but it's hard to interpret those results because what happened is the sodium distribution gets, or the amount of ion channels gets into the axons gets substantially reduced. So it's hard to answer whether it is the structure that anchored the ion channels that matters, or whether the periodicity of the structure that anchored the ion channel in a periodic way that matters. So what we're doing is to create genetic systems so that we can not do these more strong perturbations, but more subtle perturbations so that the molecules are still there, such as spectrum that can link to anchoring and so on, to anchor the ion channels, but the periodic structure is gone. So that requires more subtle genetic mutations on the molecules, and we are getting, ready to do that kind. I mean, I'm saying that we're trying to get ready to do that kind of more careful perturbation. Yes?
You label these molecules with very large fluorescent dye molecules. And I was wondering, are you worried about this influencing the structure of the molecules and the interaction of the molecules? Is that an issue at all, or is it not an issue?
It is something that one needs to be careful about. So there are different kinds of labeling approaches. And some of the labeling approaches I talked about today, it's less of an issue. For example, in the second part where we do fluorescence in situ hybridization, and those are cells that are already fixed in the first place. And then also for the protein imaging, some of the earlier parts that I talked about, there are two kinds of generally used labeling approaches. One is more geared towards fixed cell imaging. The other is more geared towards live cell imaging. The fixed one, if you actually use antibodies and so on, again, the cells are fixed to start with. So if you care about whether the molecules are still behaving, and then since it's already fixed, it might be less perturbative. But if you are linking a fluorescent probe directly to the protein molecule in living system, then we often do test whether that connection can happen between fluorescent protein genetically fused to the protein, or some tags such called SNAP and halo tags and so on, and then you link a dye to it. In those cases, we often do test whether the function of the protein in the living system has been perturbed by the labeling. Now, if you ask a different question, that is whether, not the labeling itself, but whether the fixation could perturb the structure that we study, then sometimes we also have to care about whether the fixation process has perturbed the ultrastructure that we're imaging.
Question, yeah.
Thanks for your talk. So my question is, your stone is pretty limited by the light propagation, so you cannot penetrate very deep. But the majority of the biological procedure is in the deep tissue. How can you probe most of the biological procedure?
Right, so that's a good question. So maybe just to clarify a little bit, I will answer to that, but I want to clarify at the beginning that it perhaps is not entirely accurate to say that most of the biological processes cannot be studied, because a lot of the time the light penetration depths and so on is allowing us to study not only the cell cultures, but also live tissues and so on. So very often, a lot of the biological processes are revealed by light microscopy, because STORM is just another light microscopy approach. So the penetration depth issue is the same, okay? So was that answered, was that clarified? I do agree with you that if you want to study deep in tissue, there are additional challenges. And there are a variety of additional challenges that one can think about answering with a variety of different approaches. Now, the first level of challenge comes from aberration. So very often, our imaging system's refractive index do not exactly match the refractive index of the tissue. And then with that, as you go deeper and deeper or more and more away from the surface, you get stronger and stronger aberration. And these aberration can actually be corrected by adaptive optics. So that's one way to go. Now, the second thing is, as you go deeper and deeper into the tissue, STORM or typical sensitive imaging approaches that rely on the detection of single molecules run into a trouble of signal-to-background ratio, because we need the single molecule signal to stand above the fluorescence background. If you go very deep, the background gets accumulated pretty strongly. And there you can use optical sectioning approach. For example, the light sheet, I don't know whether you have heard of that. You're sending light that look like a thin sheet, and then with that, then you can actually remove the out of focus background. So that's one way to overcome that. The third level is the hardest, and that is the scattering. So once you go deep enough, light scatters. And with that scattering, imaging quality gets very much deteriorated. Now, that there are sort of active effort, but not great solution yet in dealing with the scattering-induced problem. But these things happen deeper and deeper. Scattering, for example, aberration, you could actually use adaptive optics to solve problems enough so that a few hundred microns could be imaged. Multiple millimeters is, maybe 1 millimeter is already enough to get scattering.
Yeah, millimeter is really bad.
Yeah, so then that's, at that depth, then that scattering gets a major problem. And I mean, there are ways, but as I said, there's a lot of improvement that needed in order to battle the scattering problem.
Other questions? Okay, thank you.
Thank you.
Go from the weak scattering limit, and you have special functions that Eric and all those continuing